More recent antibiotic. It's a slightly different version, very related to termicin. They're in the same category of antibiotic. And this actually does work so far, but we have seen that there are Thailand-resistant American foul brood strains of bacteria cropping up, which is kind of some bad news because that means at some point in the future, Thailand is also not going to work. One of the other problems with using antibiotics, aside from this resistance, is that there is going to be residue of the antibiotic left in your hive. If you are having to treat for it, usually you put the antibiotic powder in a powdered sugar and sprinkle it over your hive. The bees will go and eat that, and at the same time they'll get the antibiotic. So you can, if it's able to be killed with Thailand, you can actually treat them and they can recover from an infection. The honey that they're making at that time will have this antibiotic in it, and this is not actually, neither one of these are actually good for children or developing babies to actually be exposed to. This family of antibiotic will cause bone growth problems. So a developing fetus or a child that's growing, they can have teeth problems or bone problems of growth if they're exposed to these antibiotics. So this is why currently in the United States, if you have to treat your bees during honey-producing portions of summer, you're supposed to throw away all that honey that they may have produced during the time, or only allow it to be fed to the bees and not to people, because we don't want this to be in our food supply. Let's see. So that was what I just said now. Okay, so this is the family of drug, but we'll skip that slide because it's like super boring looking. That one looks boring too, so we'll skip that too. But it was really what I just said in words. So the other thing that happens then, if you have a hive that's too far gone, like sometimes the queen will, she'll actually basically have trouble, since she can't maintain brood, she'll stop laying, or even there are enough bees that they haven't supported the colony. Sometimes the queen will leave and you get basically the whole colony gone. You come back and there's no bees, and what's left behind are racks with just a few workers that were left behind. These untreatable hives have to be burned, or if you treat them with an antibiotic and they don't recover, you basically are supposed to, within 2 weeks, in the state of Utah, we have 2 weeks to treat and see if they actually make an improvement, and if they don't, then we're supposed to burn the hives. And this is so that we can maintain the control of the bacteria, because if we leave a hive that's going to be dying anyway, that means other bees can come in and actually spread the infection in the area. Okay, so that's kind of like the introduction of the disease, and now I want to tell you about the work that we're doing. So here's the bacteria, and what we're looking at is called a phage. And a phage is actually a type of a virus, but it will only infect bacteria. So it's kind of dark on this screen, but there's like a head here and a tail. And this picture here is the side, a close-up of the side of one of these bacteria. And all of these little white round balls with the little tails, these are individual phages that are now attached to the bacteria to kill this bacteria. So I think I have right here a little video. It's also really dark, but I don't think that's gonna, oops. Maybe, go back. Where's my, let's see if it will play, and if the sound will work.
At BYU, we're doing research on phages that will infect and kill American foul brood. Phages are viruses that infect bacteria. Phages are the most abundant life form on Earth. There are phages in the soil, in the air.
Kind of look like a lunar lander a little bit. Cats with a long tail and these spindly legs that come out and bind onto the bacteria. So when a phage attaches onto a bacteria, it'll tuck down and embed itself in the membrane.
And it will inject the DNA information from the phage into the bacteria, and use its own DNA copies inside the bacteria to make more phage progeny. When the phage progeny develop enough, they can actually break open and the bacteria is dead and it releases thousands more phages. At BYU, we are sequencing the DNA so we can see what are the genes that are in these phages.
Somebody gave us a frame of foulbrood that they got from their hive.
The larva has been basically eaten by the bacteria and turned into a very soupy, stringy mess. The phages that we want to find are phages that will infect and kill the bacteria that causes American foulbrood.
Okay, so that's kind of the introduction of what these phages are. And here's a little cartoon depiction of that bacterial cell and the phage attaching and then putting its DNA copies so that it can make phages inside the cell and then break that bacteria open. So it's interesting because phages have been used to treat bacterial infections already in the United States. There are phages for some plant pathogens. There's a tomato blight that you can actually buy phages to spray on, and it's usually in a liquid form, and then you spray this. This is spraying a tree, but they also, you can put some of these phages in a crop duster and then spray the tomato fields. I know that's corn, but sorry. But in any event, you can actually spray them on and kill the bacteria and have healthier plants from it. So the other is that we also have phages used. You've probably been eating some phages on your meat because they use one called Listex that's approved that is used to treat meats before they go out for sale in the grocery store, and that kills a bacteria which is called listeria. So this will prevent food poisoning from listeria. So we have phages that are currently in use for some of these. So there's the plant one. So the beautiful thing about phages is that they're very, very specific for the bacteria that they infect. They cannot infect people, they can't infect animals, they can't infect plants, they can't infect anything but a bacteria. And then they can only infect that bacteria that they're very, very specific for. So the phage that is used to treat the bacterial infection in tomato plants is a completely different phage than that's used to treat against the listeria bacteria in the meat supply. And so, and these, because they are abundant and they are everywhere, these are considered organic. This is part of nature, one of nature's ways that we see it actually fighting bacteria. And that means if we do a treatment of phages on a beehive, this is actually an organic treatment to treat for the bacterial infection, and the honey is completely safe to eat, and we don't have the same problem that the antibiotics have of getting in the honey. So in the lab, we have bacteria that we've isolated. There are some bacterial strains of American foulbrood that we can buy from this company, ATCC. They provide some specific ones, and these ones are quite old. They're from about 30 years ago. And so we actually decided to get field isolates of what currently is happening in the United States. And of course, we started out here in Utah. So here's some places that we had beekeepers donate samples, and then we would take them to the lab, and they were just from healthy beehives, and we would find those spores that were there, not necessarily make it an infection, but we'd activate the spores and grow the bacteria in the laboratory so that we could have a bunch of different types of American foulbrood, that bacterium that causes American foulbrood, and see what it is that we're seeing happening in infections in Utah. So we'd have healthy larvae or bee debris. This is like outside the hive where they throw, they're dead off, and you can have some on the ground. Or we did get samples from infected hives. So we would grow the bacteria in a culture, and then the cool thing is, so here's a petri dish, and what we do is we'd spread the bacteria so it basically makes this lawn of bacteria in the petri dish. And here we gridded out on that lawn where we put a drop of a phage in each of these different places and see which phage could actually infect bacteria. And when it infects the bacteria, it will kill the bacteria in that area. And that's why you see there's actually clearing spots here where the phage landed in, and it actually is killing. So it was pretty exciting that we could start finding these phages that would actually kill American foul brood bacteria. So this is another map of like the different places where we found phages, again from samples from beekeepers who sent in samples that we could test and see are there phages. So one of the hypotheses I proposed is that there must be phages that are happening in the beehive. And that's why sometimes you have a person and they have a hive and it does great and it never comes down with American foul brood and not very far away, or maybe even within the same apiary. You can have another hive that gets sick and then you're fighting AFB, or you have to get rid of that hive, and the other one does fine. So these naturally occur. So we take samples from healthy bees, from healthy hives, and from also sick hives, and we can find this phage. And this is like the nerd science picture that I have to show you because my students were so excited about doing an electron micrograph of the bacteria with on right down here on the surface here, yeah, right down there. If we enlarge it, there's actually a picture of our phage attached to it. It was really super exciting. So, you know, that was like our science stuff that we're doing. But we do take and we sequence, we take these plaques and we sequence them. So we look at the DNA patterns and we try to look at what proteins that they make and see how we can actually learn more about American foul brood by studying these specific phages, and also to see, you know, what phage might be really the best if we were going to spray it on the bees to treat the bees for an infection. We've basically been leading the world in doing phage research at BYU. So this is the phages that we have so far, and we've teamed up with some other schools. So UNLV has been doing phage research for a few years, and they have quite a few isolated, but not necessarily sequenced, and we're helping them with that project. And then the North Carolina State University has some phages, so we've been collaborating with them as well. And there's a group in Portugal that has one phage they published. We've seen that they have it. I haven't never contacted the people there. And there's also a group in Germany that's doing some research. So this is very new work that's happening, and it's really exciting to be a part of this movement and seeing what can we actually do to help control and manage American foul brood by using these phages as a weapon. So this is, again, an example of some of the work that my students do, which is not just work with bees, but really look at some of the genetics here. We look at a comparison of how these phages are related to each other. So that's kind of a fun part. Let me show this next little clip here.
The way that we isolate specific phages out of a sample that may contain hundreds or even millions of phages is we mix the sample with the bacteria that we're trying to isolate. If there are phages present, they'll amplify. We put the bacteria on a petri dish and then we'll add our phage sample on top. And then if we can see places on this petri dish that are clear, they have no bacteria, we know that a phage is responsible for that, and we can continue working with that spot and isolating the phage further. By isolating them from the environment and then using them for a specific purpose, it's an organic treatment. Because we haven't changed anything, we haven't modified anything, we're just using nature's natural bacteria killers to fit our specific purpose.
It's exciting for us at BYU to do phage research, especially on this particular bacteria, because this is clearly a worldwide problem in honeybees. We've published 5 full genome sequences, and so far only one other genome sequence for a phage against the American foul brood has been published. The phages are all in solution, so we prepare a sugar water solution of the phages and spray it on the beehives. When the bees get sprayed with sugar water, they stop and clean themselves. This is really useful because the nurse bees that get sprayed are going to clean themselves, and then they're also going to go and feed the larvae.
We're able to go to a beekeeper and see their affected hive and apply our phage treatment. We could see noticeable improvement in the hive, which was really, really satisfying to know that something we developed in the lab is actually working in the field.
Let's see. So we found some other phages. They're pretty cool. We got these ones with like a little oblong heads, and some of them with the little round heads. And you know, this is like some little round pieces and stuff. So we have these new from samples. These are from Wisconsin has been shipping us some samples, and this one was isolated in 1980. So when I started publishing work on these phages. The original first ever phage that was found in 1980, the researcher who was just about ready to retire, he saw my paper and contacted me and said, hey, I'm excited that you're actually